Translation is basically the production of amino acids, and this production happens in the cytoplasm of the cell. Remember from the first video, amino acids are the building blocks of protein. They make up proteins. So in this video, we need to somehow create a bunch of amino acids in order to build a protein. So going into translation, the very first thing that happens is the binding of the small ribosomal subunit to the messenger RNA. This is the small subunit of the ribosome. It's also called the 30S ribosomal subunit. For this binding to take place, there are initiation factors that have to bind to the cap region of this messenger RNA. First, recall that during the post-transcriptional processing, the cap is added to the 5' end of the messenger RNA molecule. Oh, and this is the 5' end. This is the 3' end. Other initiation factors will also need to bind to this small ribosomal subunit to start the binding. So in other words, these initiation factors, which we don't see here, are the things that push the stop button for this small subunit ribosome to attach to the messenger RNA. Here you may notice there is a codon. This is the start codon. It's made of the 3 nitrogenous bases AUG, and it always codes for methionine in eukaryotes and archaea, and also in some bacteria as well. You might be curious and ask, what exactly are the ribosomes? Well, ribosomes are basically the main component that is needed for translation, and they can be found free in the cytosol. Some of them are attached to the rough endoplasmic reticulum. You can simply think of them as machines that have the ability to produce amino acids and thereby forming polypeptides. And of course, the tRNA, transfer RNA. Without transfer RNA, the process of translation just cannot function. tRNA carries the appropriate amino acid to be added to the growing polypeptide chain. Here, the tRNA or transfer RNA is then going to bind to the messenger RNA by the law of complementary base pairing. Once the tRNA lines up perfectly along the messenger RNA, the large ribosomal subunit. This is the large subunit of the ribosome. It's going to step in and bind onto here. After the binding of the large ribosomal subunit, the initiation factors, which again we don't see here, are then going to be released. So what's happening up until this point is known as the initiation process, or the initiation of translation. Now you see inside the ribosome there are two areas, this part here and this part here. This part is called the P site, where the first tRNA over here with methionine is positioned, and this part is called the A site. Following initiation, another tRNA will occupy the A site. So this tRNA will occupy the A site of the ribosome according to the complementary base pairing between the anticodon of the tRNA, so this is the anticodon, and the codon of the messenger RNA. So the messenger RNA codon will be A, C, G. Once the two tRNAs occupy the sites, the enzyme, which we don't see here, called peptido, transfers. and peptido transferase will start to catalyze the formation of a bond called peptibond between the two amino acids. The first amino acid, which is always methionine, is then going to be released from the tRNA molecule on the P site of the ribosome. The free tRNA molecule, which is the tRNA without its amino acid, leaves the P site of the ribosome. The ribosome then moves down 3 bases, or 1 codon, down the messenger RNA, placing the second tRNA on the P site of the ribosome. While that's happening, the next charged tRNA over here enters the A site, which will bring in the third amino acid to be added to the polypeptide. So this process will continue until a termination codon is reached on the messenger RNA. At that point, the polypeptide is released, and the ribosome and the messenger RNA dissociate. One thing to know is that for one given messenger RNA molecule, there are actually more than one ribosomes that translate it, and the ribosomes are doing their job at the same time to create a unique polypeptide. Polyribosomes are what we see when multiple ribosomes are working together on a messenger RNA. This photo, a colorized micrograph, shows what it looks like. We have a ribosome here, another one here, another one here, and it's hard to see, but the messenger RNA is actually over here. You can see the ribosomes are on the messenger RNA by the formation of the line, and they are working together as a group, polyribosomes.